This video is the cell cycle, introduction to cyclins and Cdks. I want to point out just briefly that the cyclin and Cdk that you see listed here with the M in front of them, those are xenopus and non-mammalian names. So for the cell cycle, there's two types of cell division machinery. And they're listed here. So the first one is the cell division machinery itself, which will make new or duplicate things. It also places items in the correct locations. And so I have a symbol for this machine over here, which looks different than the machine that's over here. The other part of machinery that is involved with cell division is the cell cycle control system. And so basically what this does is it coordinates the cell division machinery by activating and then deactivating key proteins and protein complexes. This is going to allow it to go through different checkpoints and to progress through the cell cycle. In particular, the regulation events that take place are those for DNA replication, mitosis, and cytokinesis. And the primary way that this happens is through regulating the activity of Cdks. So you could kind of think of this as the cell as a big factory, and so there's two different types of machines that are working to allow that cell to divide and to progress through the cell cycle correctly. There's going to be a variety of different workers and specific workers that work with one set of machines versus another set of machines, and so someone needs to coordinate that. So this could be, for example, the foreman or some of the other individuals that are going to coordinate the shift changes through activation and deactivation. And so in this case, the timing of the shifts can change depending on what work gets done, unlike in a normal factory where there really are specific start and stop times, at least as far as I know. Our focus is on the cell cycle control system. So this shows figure 18-4, and this is one of our take-home messages, that there are two main characters for the cell cycle, namely the cyclin and the Cdk. Each of these operates as a pair, so there's a variety of cyclins and a variety of Cdks, and they always work together. As you see some partners down here, some famous duos, which always work together. And their role is to regulate the passage of the cell through a certain checkpoint. It's the same basic concept for all of them. Cyclins have no enzymatic activity, and they must be bound to their proper Cdk for them to be active. The most important thing to know about cyclins is that their concentrations vary throughout the cell cycle. And this is one of the primary determinants of whether the checkpoint will be passed and whether the cell will go on to the next step in the cell cycle. Cyclin-dependent kinases, or Cdks, these can be phosphorylated or dephosphorylated by other enzymes, which we don't see here. And you can see in the picture here that the Cdk has the little extra tiny red lines, and so that indicates that in this point it's in its active form. And that's due to the binding of the cyclin. As a reminder, the Cdk is the same protein whether it is phosphorylated or not. That's just a small covalent modification. It may change some of the shape of the protein, but still you have the same protein sequence. As a reminder, that's also a take-home message. So let's talk a little bit about regulation. So how the partnership is regulated is through two things. First off, through the Cdk's phosphorylation state, and secondly, by the presence of cyclin. For what we'll talk about, especially right now, the key part is the cyclin concentration. And this is a direct relationship that when there are high concentrations of the cyclin, then there's high activity of the corresponding Cdk. This is figure 18-5, and this shows that concept in a graphical form. And this is our big concept here, also take-home message, so that the cyclin cycle over time as far as how much is present, and the Cdk activity corresponds to the cyclin concentration. You could also think of the Cdk activity cycles over time. However, the Cdk protein is present either way throughout the cell cycle. Okay, so I want you guys to use the figure over here and to look at this kind of spoiler cell cycle, which I had at the end of the take-home messages and on our first lecture. And so basically I want you to sort out which checkpoint is shown and which duo is shown. So to figure out which checkpoint is shown, look at the top part here and see where we're getting the activity and what's the transition point. So notice that we've got the CDK activity, and that's happening right at the tail end of interphase and going into mitosis. So if we look over here, that's the G2 to M checkpoint. As mentioned at the beginning of this video, the M, CDK, and M cyclin, those are the xenopus or non-mammalian versions of the CDK and cyclin. So those names are not the same names as you see on the spoiler reference over here. So if we check out that G2 to M and see what the names are of these cyclins, the cyclin and CDK, it's actually cyclin B and CDK1. To clarify, those are the mammalian names on this reference, and the non-mammalian names are shown on the figure over here on the left. And just to also point out, Xenopus is a frog genus, and so that's where a lot of the initial experiments were done for characterizing the CDKs and cyclins. And essentially what this means is that cyclin B in mammals is very closely related to M cyclin, and we can tell that by the protein sequence. And likewise, CDK1 is very closely related to M CDK. And so these are more closely related to these cyclins than to any other cyclins, and likewise these two CDKs are most closely related to each other compared to any other CDKs.